will take the floor. Thank you. Good morning. How are you all doing? Thank you. We want to talk about biologic nitrogen, new tools. With Alejandro, we prepared this space to come back to basics. We will speak of cycles and understand why we're here, what we are doing, and in particular, where we're heading to with such an important tool. Remember, nitrogen is a vital element for the functional performance of the plants, physiological processes. So much so that in the planet, 78% of the air is composed of nitrogen. So the main reservoir, we come across it in the air all the time. But the problem is that it is not usable in a direct way. So we have a great reservoir, we have great potential, but the means by which it goes from air to the plant and turns into growth and production, those are very different mechanisms, very complex ones. In the nature of that complexity, it comes from very complex biological processes with microorganisms responsible of the functioning of systems so that plants grow, organisms in the interaction with plants and other greater organisms can make sense to the production system, which is what we're interested in. From nature, we make a system. Remember that. In that complex nutrient cycle, that cycle that has a great reservoir of nitrogen in the atmosphere, microorganisms become essentials. They're not just part, they're essential part. Without microorganisms, there is no entry of the nitrogen into the cycle, so we don't have the vital element for plant function. In that process that has been studied at industrial level to fix air nitrogen in fertilizers and to incorporate it in plant nutrition, we see that each one of these squares, maybe I cannot read all this process, it's kind of blurred out, but I see that it links to specific microorganisms that are nitrogen fixing and nitrogen binding and transforming. They make that nitrogen from the atmospheric complex form. It is simplified and it is made available and reactive so that plants can incorporate it. This is ammonium nitrates that plants incorporate and use. Out of the total nitrogen available in the air, of that large amount that we see, this is statistic data worldwide, nitrogen fixed and incorporated are 290 million tons of nitrogen. Some by non-biological processes without the intervention of microorganisms, but with a macroorganism involved, which is the industrial process, incorporation through fertilizers. The rest, that 60% remaining 60% of nitrogen incorporated through biological processes, out of that, a small amount is by sea microorganisms, mostly found on soils through biological processes that we know they occur non-symbiotic fashion, that is without a particular species in between, and in a symbiotic fashion, biological symbiotic fixation, most mostly by legumes. So we have here a cascade of different amounts, and that's why the biological use in fertilizers and the biological decision through production systems is so important. How plants intervene. How do plants intervene in this nitrogen fixation? There's symbiosis, where two organisms coexist, help each other, and aid nutrition. The example here is legumes. There's a synergy, incorporation of nitrogen straight into the plant. Others are by association, where the microorganisms take the exudates around the roots to grow, and they can capture nitrogen from the air, transform it, and that non-specific form nitrogen is contributed to the roots of that species and others, and that free life, they fix nitrogen. That is, they can be in water, in soils, and they can capture nitrogen from the air and transform it to put it in the soils and cycle it through nutrition. All that is for life on soils. No exam, but to give you some references, in each one of these categories we have identified microorganisms that contribute air nitrogen for their life and contribution direct or indirectly to other plants. Some of them creating structures, some endophytes, some in free life around the root, and others within the plant, the endophytes, for example. So we'll stop in these three categories. All of them need nitrogen for life, and throughout the life cycle, nitrogen is essential for the function. The plants need nitrogen to live, and that's where things become more complex. This is brand new. It's a recently published review on why it's important to have nitrogen fixed. Is it a microorganism? Is it a species? Is it the environment location? Is it the region? No, this is a very complex review saying that biomass production, that is, the growth of plants, make the amount of nitrogen fixed and incorporated to the planet. So we have microorganisms as species in different positions, and the crop growth condition as a driver of that improvement. Out of this study, it's important to highlight that year after year we see information showing that nitrogen fixation occurs because plants grow. That's the order of factors. Everything that contributes to growth, nitrogen offer matches our capacity to improve plant growth. And biological nitrogen refers directly to a definition: nitrogen assimilated by plants through living organisms. That assimilation is the incorporation of nitrogen in a system when, because it was wanting or limited, we add fertilizers, we add legumes that need to grow to fix nitrogen, and in the end, the growth process makes sense to incorporation of biological nitrogen in the systems. We broke it up into biological nitrogen is the one fixed through legumes directly. Air nitrogen becomes simpler so that a specific species can make the most of it. And then we'll talk about the derivate, that is, derivate nitrogen incorporated by an organism. captured in its structure to function and grow and accompany plant growth. That plant growth captures nitrogen per se, and we are incorporating it to fertilizers, mineralization of soils, and others. Biological tools and technology integration is a key optimizer because either applying species that generate symbiosis or introducing microorganisms that provide efficient plant growth. And if it is efficient, it incorporates nitrogen for functioning of the system. This is a study on wheat crops in Australia, and it shows the importance of inoculating with microorganisms. This is because if you see the red line, the incorporation, and the blue line that points out the natural condition, inoculation accelerates a natural process. So if you add a microorganism, you introduce a favorable condition, so the positive characteristics are expressed. And that led to a formulation that Alejandro will share later. It just points out the identification of rhizobes to favor plant growth unspecifically, but they fix nitrogen. In the case of legumes, we know that when we inoculate, we increase air nitrogen capture. We know that even in soy, inoculation is an important mechanism to explain this. The greater the production, remember, production is a synonym of growth. If growth is the factor aiding nitrogen fixation, then when you lack biologic fixation, yield is stable, but if productivity increases, growth increases, the contribution to the amount of nitrogen coming from the air increases. So biological fixation is the tool that responds to growth improvement that we are used to when we talk about legumes. But what happens in non-legumes? We incorporate microorganisms that improve growth of the root, more biological nitrogen incorporated. It allows to manage biological processes. It grants stability in production. These are results of corn and wheat with Azospirillum. It's always by incorporating species that are present, but the location at the root improves growth condition or nitrogen nutrition. And also knowing that there are other microorganisms that can grow as endophytes within the plant, we are exploring growth improvements for crops that can be explained because of a greater nitrogen uptake, not by substitution, but by greater efficient in the use of an essential element for plant growth. This is leaf application on corn and soybean. It's nitrogen supply during growth, greater efficiency, greater contribution of nitrogen nutrition. And in the case of soy, because it's inoculated with biological fixation directly from the air, depending on the formulation, the green or the blue one, we see improvements in efficiency. this is in the reproduction phase or the growth phase. So the improvement with microorganisms associated to nitrogen fixation improves the crop nitrogen economy. The application on the seed and on the leaves. Well, each individual should receive the inoculum adequately. On the right side, the formulations and the active ingredients are different. It depends on how the formulation, application, and growth stages are. That's for effective use of this. There's broad experience on rhizobia, showing and validating that we are incorporating nitrogen that complements and improves growth conditions. The implementation of these new management alternatives to improve biological nutrition allows a better understanding of all this. This is just the introduction, so that now Alejandro can tell you nutrition studies, how they assess technology, and what results it is yielding in the field. So Alejandro, it's your turn now.
Verde.
La presentación la
Muchas gracias, agradezco.
Thank you very much. Thank you, Aapresid, for this privilege to present biological nitrogen and allows Rizobacter to showcase how a company can continue innovating with technology and to look for sustainable agriculture. Nitrogen, as explained by Martín, is essential, is a macronutrient. After water, probably is the most limited that a crop has to develop, to grow. It has many essential functions as a component of amino acids, enzymes, proteins, part of nucleic acids, intervenes in photosynthesis. So if we have a deficiency of nitrogen, for sure we will see a limitation in the production of our crops. And what happens? Well, we do have a lot of nitrogen. The atmosphere has 78% of concentration of nitrogen per square meter in the surface of the Earth. If we look up, we have 4 tons of nitrogen, so we have a surplus of nitrogen. The soil as well, the organic part has nitrogen, but the roots cannot take advantage of it. It needs to be inorganic as ammonia, as nitrate, so that the plant can take advantage of it. That happens through microorganisms. What enabled genetics of the crops is to grow in yield. In the management, the genetics led us to being able to explore high yields, and the only way to supply this nitrogen need by the plants was with the use of chemical fertilizers. So chemical fertilizers enabled us to have huge yields compared to 50 years ago. Think that during the last year, in 1960 to today, it is estimated that the amount of chemical nitrogenized fertilizers went 9-fold, and for 2050 is going to double-fold what we used to today. So the use of the chemical fertilizer enabled us to to solve hunger and to have a better yield. But we need to have a sustainable agriculture because we have an environmental impact in place. And in 30 years, we are going to duplicate the use of this nutrient through chemical fertilizations. The environmental impact will be worse. So what is the environmental impact of nitrogen? Well, there is a lot of research showing that, and we all know, that in order to manufacture an nitronized element has a very high energetic cost. 50% of the entire energy in agricultural production is for the production of nitrogen. And we also speak that natural gas used in the agricultural system, in the entire agricultural system, 50% is contributed for the production of nitrogen. That leads to a lot of greenhouse effect for the manufacturing of chemical synthesis nitrogen, and when we use in agricultural nitrogen chemical production, we also increase the production of nitrous oxide because it has more potential for greenhouse effect than the other one. So we need to reduce the emissions thinking in 2050. Let's not forget that nitrogen, out of all the chemical fertilizers manufactured in the world, 50% is used by the plants, 50% is not. So if it rains or if we are in an area that it has sandy soil, that nitrate can be washed over or can be run off and get to water flows affecting ecosystems, water ecosystems. So nitrogen has an impact on the environment apart from being used as a chemical fertilizer and its yield. So we look for more sustainable tools. As mentioned before, the biological fixation of nitrogen is a sustainable tool and responsible for the fixation of nitrogen in the earth of 60% of the total amount. We estimate 150 million tons per year fixed by the nitrogen fixation. Can be freely or by symbiosis. We all know that symbiosis in the case of soy, that relationship where they are both benefited, and that happen in an organ called nodule. And then we have the free form. And here we start to explore new types of biological fixation of nitrogen. That's something that happens in nature, and something that happens in the philosphere. What is the philosphere? The air part of the plant, the stem, the flowers. So everything that is under the soil, the root. And there you have microorganisms with different functions. Bacteria, virus, algae, fungi. The first habitat in the world in terms of microorganisms is the soils, and the second are the leaves of the plant. So that is the second habitat of microorganisms that we get to know more and more every day. And there we find a lot of bacteria, different genders. We have Rhizobium, Mesorhizobium, Nasopyrillum, Bacillus, among other microorganisms and other bacteria that live on the leaves. These microorganisms, at the same time in the philosphere, can be epiphytes. They live on the surface of the leaf and they develop there, or endophytes. They have the ability to enter the leaf and to live inside the vegetable tissues. So within this bacteria group found in the philosphere, we find also another group that is called methyllotrophic. And which are the methyllotrophic bacteria? The ones that are able to use as energy source a composition of carbon that can come from methanol or methane. That is a process freed by the photosynthesis. We had the Methyllobacterium method, and its energy source to develop is the methanol, and this community is known, we know now that we have different types of methylobacterium, and in their genetics component they have the NIF gen, the ones that are able to synthesize nitrogenate. That is responsible for doing away with that nitrogen and to have a natural fixation and provide ammonia to the plant. So here we have an impression of a leaf, and that leaf was inoculated with methylobacterium, and that impression took place on methanol, and then it was removed. So what we can see is that those bacteria that were in the middle of the crop started to feed from that methanol and started to develop. In Rizobacter nowadays we are working along these lines and working with this technology. We're developing a product with 3 bacteria, 2 of the Methylobacterium type, and 1 gem that is called Aapresid globiformis. These were 5 years of development to choose which were the Methylobacterium that were efficient, not only fixating nitrogen, but also resisting on the leaf. They must be efficient under several conditions so that we can find a good answer with all the crops. They have to be resistant to different conditions. This is a foliage product for desiccation, for many adverse conditions that are placed on the leaf. So they are applied in the phyllosphere, and they can enter through the stoma or the gaps in the cuticle to the tissues and colonize the stems, the leaves, and they can even found in the roots. They complement each other, and they can adapt to different crops. It's not just for one species, but it can adapt to many. So how do they live in the leaves? And this is important because you think about the microorganism, we drop them on the leaves, it's exposed and it's complicated. Well, these bacteria have 2 strategies. They generate a biocoverage through polysaccharides that enable them to defend from desiccation. And then when violet ultralight, these are carotenoids that are synthesized, and the bacteria with those carotenoids can absorb the energy of the ultraviolet radiation, and they can produce antioxidants to remove these free radicals, and if they don't have it, then the ultraviolet radiation will kill the bacteria. So that is part of the natural evolution of these mechanisms, these microorganisms. They have done it for years and years and adapt so they could live and develop on the leaves. We find now a product that we will launch to the market with several benefits. Bionutrition is one of them. We have been working to understand how this bacteria works, and one of them is the nitrogen fixation, a green, clean nitrogen that is leveraged in its 100%, is used by the plant. We're not going to have any loss. Another thing we understand about these bacteria is that they have a biostimulant effect. They produce hormones, or this creates or enables the plant to increase its foliage expansion. When we increase its foliage expansion, we are also creating from the plant to the roots a higher demand of nutrients and water to enable the photosynthesis. So this growth hormones is not only for the foliage, but also creates a better radical root growth. It also has an effect effect, and it's a natural competitor against those microorganisms that can be pathogens, because here we have a product with a high concentration of colonist formers. So when we apply this product on the leaf, we occupy a big space, and we are enabling these microorganisms that can develop very fast. They occupy spaces that can be occupied by other pathogens. So it has good impact on that side. And this is all green because we are providing nitrogen to a crop. We are not manufacturing it, and that also creates a high economic cost. So we have had several essays in different places. This essay in France, or this test sampling, we show the X control, which is the normal doses used by a producer, a 100% control. We saturate the corn with nitrogen, and then we have the Rizobacter product complementary to the nitrogenized process, and we have an answer, and the answer covers almost 100% of the total amount of the nitrogenized production. Another essay by Gustavo Ferrari shows that with control of 100%, one doses of 270 kilos of N versus control N, which is 300 kilos of urea, is what is used by the producer. And when we add these to what is used by the producer, we have an increase of 10% in the response. We have tried this with soy. We found more than 300 kilos of response. We are trying with other type of crops. What we see in the case of soy is that there is not a problem with this type of bacteria. We are working with potatoes, and on the fertilization by the producer, for us is a technology that is complementary to the one of the producer, where we have different types of responses: biostimulation, using better in the fixation of nitrogen. Then we are providing this green and cleaner nitrogen, and if we improve the efficiency of the use of nitrogen, we are also improving the absorption of that nitrogen that we use as fertilizer for the soil. So those are 47 years of work of Rizobacter working in biologicals, and the idea is to continue innovating, working and developing solutions for the producers that are complementary to each other, and to have a more sustainable agriculture in time. Thank you very much. Well, we can have now time for questions, for exchange. There is a QR here on screen. You can send us your question. And in the meantime, I have a few questions for Alejandro to know more about this new development. You spoke about the mechanism through which these microorganisms that we are applying in an endogenous way, we can apply them in a foliage way. They are going to work endogenously, and they are going to improve the use of nitrogen. They provide nitrogen. So from the application conditions perspective, how are you positioning this? What are you doing in terms of methods of application, environments? We are working with live microorganisms. Yeah, here the recommendation is the early application, as early as possible. In corn, for up to 15 days we measured, and we were able to find this type of bacteria in different parts of the plant. So colonization of the plant is quite fast. We have a big amount of chemical products that are compatible, and we didn't have any problem. This is a bacteria that is quite extensive and highly tolerant and resistant to different types of chemicals, and we don't have any restriction in terms of application. Can be applied during the entire day. We need to be more careful what happens with the relative humidity. We do not consider an application under the 30% of atmospheric humidity, but the producer rarely applies a product with a low relative humidity. And this is a liquid product with organisms that are alive in the liquid to fulfill its function. I have two questions. Did you mention anything? You mentioned something about hormonal processes and the nitrogen release and fixation. Can you tell us a little bit more about this mechanism and how it would work, this hormonal process? And in terms of nitrogen, if you see that there is a replacement of urea, or if you have any recommendation. In terms of hormones production in the metabolites produced by this bacteria and this symbiosis that it has with the plant, so all throughout these years, the plant had restrictions in nitrogen and other limitants, and it created this symbiosis internally where they are both benefited. In the Metallobacterium uses as carbon source the methanol, and Aapresid does not use methanol, use sugars that come from the vascular of the plant. So they use these hormones as synthesis, and there you need to find a biostimulation effect on the crop that comes from the hormonal side. In the case of, I don't remember the other part of the question, the replacement of, we are working as a complement. We are not increasing the doses of the producer. And we try to manage this very carefully. We have work that is mentioning 5 to 20 kilos of fixed nitrogen per hectare, what is showing the bibliography, but we are working to have more specific data. But that is very variable because it depends on the crop, the conditions of application, the environmental conditions, how much a bacteria can fix. However, how we can replace these, we are not going to go with this replacement line and what the producer does, because in Argentina, what we find many times is that the nitrogen doses is not enough compared to the yield potential of the crop. Perhaps if you go to Europe, it's the opposite. They have an overdoses of nitrogen, and they can work with that replacement. We are more along the lines of what the producer does, and probably we will have a better response or a more complete response. There are a lot of questions. We will try to encompass them. Many questions about moments of application, thinking about the crop. How long it takes to initiate the nitrogen fixation, activate the microorganism, and provide a benefit for the crop? Do we need to apply it again? What happens when we reapply? Or for how long this benefit lasts? Once we apply it in early stages, we estimate that in summer crops, in 15 days, we have the plant fully colonized. The main place where this Metallobacterium find is at the leaves, but they colonize stems and vascular vessels and roots. Especially the bibliography shows that nitrogenous is found in all those areas. Once the bacteria colonizes the plant, you will have it during the entire cycle. The production of nitrogen, the fixation of nitrogen that this bacteria can have depends on the photosynthesis rate, what Martín mentioned before. What happens with this growth rate of the plant? If the plant is not growing enough and the growth rate is higher, the photosynthesis rate is higher as well. And if we have more photosynthesis, we have nutrients, we have water, and probably we will have a higher production of methanol, which is the source of energy of these bacteria. So we will have times for more growth where this bacteria, if it has energy available, probably those were the moments where more nitrogen can be provided. But nitrogen can be provided all throughout the cycle. When the plant is going to use it 100%, we are not going to have a loss of nitrogen because it is fixated internally in the vegetable tissue. You spoke many times about the moment of application, early stages, concentrations, many questions related to in that moment in before. Can we think about more localized applications? Have you tested that? And if you tested that, can you reduce the doses, decrease the doses? And those applications are affected by the volume of water that you apply. For doses, we are in 500 square centimeters per area. I do not recommend lower doses than that because we have been working on that, and that was the standard that we found. If higher doses, the lower doses is not enough. Then for broth, we have a broth of 40 liters per hectare, but if you have any client with 40, 100 liters of water, it's going to be better, of course. Well, we need to try first to see half a liter per hectare, and then to have a good quality of application. A good application quality is to use a good peak, to have this impact that it can water as much surface as possible so the bacteria can have more contact with the leaf, and then to add a coadjutant. We have 2 coadjutants of Rizobacter, and those are going to help us to break the leaf, and that broth can have a better penetration of the bacteria with the vegetable tissue. Speaking about viability and the growth and the bacteria, questions related to compatibility with other products applied in the same broth, or compatibility when it is applied, when a product is applied in a sequence, like a fungicide or a pesticide later on. What are your recommendations? For compatibility at broth level, we have a list of chemicals with which we have tested and we found no problems. What we need to analyze, and that is pending, is what happens when the producer mixes many products, 4, 5, 7 products in the broth. So what happens there is that we find 500 different mixtures. So we are working and developing these to have as much information as possible for fungicides, pesticides. If we apply the product, we have these bacteria colonizing in 15 days the air part and even getting to the roots of the plant. When we apply a pesticide or a fungicide, then it's going to be difficult that they have a negative impact. We have essays with wheat where we apply the product and then the fungicide, and we didn't see any impact or any loss in terms of performance. When you apply this product, does it have any additional benefit in terms of pathogens control, sanity? We have not measured that. From the bibliography available, we have seen that there are works carried out showing that Metallobacterium can become, in a natural way, a competitor for a pathogen, but we have not been able to test that if it improves the crop in sanitary terms. Leaving corn aside, we have many questions about application in intensive crops, especially those leafy products.
Not personally, they might be internal trials, but I only have it on potatoes. Need to work on that. And how about on barley over wheat? Do you know if on oat, it can provide better coverage in early growth. We haven't tried it, but thinking of an answer, we always try to improve things with the service crop. We improve the service crop too, but if we improve the service crop, regardless of how, it will have a good result on corn, on a biostimulant, a fertilizer, whatever. And we have two questions. How many kilograms of nitrogen is equivalent to what you estimate by this dose application in corn crops, and how does it cost? Price, we haven't commercially launched it yet. I don't have the price. And nitrogen fixation, we assess that in the bibliography, 5 to 20 kilograms per hectare. We are assessing that with marked nitrogen to see how much the bacteria can fix. And we have acetylene reduction trials to see how much these nitrogen bacteria are fixing. But that's variable. It depends on the crop growth rate, it depends on the crop, the growth rate, the genotype of the species, environmental conditions. All right, no further questions. Thank you so much for your attention. There's been many questions, broaden our knowledge, and going back to source. Nitrogen is an essential element for our planet. It's the most abundant element, but it's the most complex to incorporate. And the crop has the tools to acquire it. It is proportional to the capacity to grow, improve. So we need a synergy with natural biological processes. Not limited by the species, also finding a way to express it continuously for nutrition and yield improvement. Thank you so much. We hope we have added to your knowledge with new biological technology. Thank you.